Hello, and thank you for coming to my talk. My name is Kiefer Williams. I'm a PhD student in Sam Price's lab at Clemson University. Today, I'm excited to share with you some of my preliminary data investigating the effects of herbivory on tooth complexity in damselfishes. So when we think about teeth, I think it's reasonable to consider them as essential to understanding vertebrate evolution. As a direct interface between predator and prey, they serve several critical roles. They're often the first structural contact the predator has with the prey item. Therefore, they're important to understanding form-function relationships. Take, for example, the teeth of this seal, used for filtering krill from the water, or the teeth of this alligator, which can be used for crushing a crab. Teeth also fossilize well, making them essential tools for inferring diet in living and extinct species, such as in the horse premolars and molars on the left, or the extinct mammoth molar on the right. So we can see that teeth provide a better understanding of evolutionary history and ecological diversity of vertebrates. One important component of tooth phenotype is tooth complexity. Recent advances in quantitative methods based on 3D imaging and analysis techniques have shown that tooth surface complexity can be reliably measured to test for correlations with diet in mammals. Several of these studies have consistently found that across carnivorans, rodents, and chiropterans, herbivorous dentitions are consistently more complex than either omnivorous or carnivorous dentitions. More recently, patterns of tooth complexity have also been examined in tooth-bearing saurian reptiles, with a similar pattern emerging. Herbivorous reptiles generally have a higher level of tooth complexity than either omnivores or carnivores. And so that got us thinking, well, what about fishes? Fishes, of course, make up nearly half of all vertebrate diversity and have more dental variation than any other vertebrate group. That said, for this talk, I will focus specifically on just one group of fishes, the damselfish. Damselfishes are a large family with a well-resolved phylogeny, seen here. They also exhibit a wide range of body size, behavior, and diet, numerous dietary transitions throughout the clade. However, there has been no work to date that has investigated patterns of tooth complexity in this group. So, using the damselfishes as our study system, we set out to investigate the following hypothesis. Tooth surface complexity varies with diet in damselfishes. Specifically, we make the prediction that benthic herbivores will show the highest levels of tooth surface complexity, thus establishing a continuation of patterns found in terrestrial vertebrates. For our study, we micro-CT scanned a total of 50 damselfish species at Rice University. Once scanned, we used the free and open source software 3D Slicer to segment out the left-sided dentary of each specimen. And for our study, we also only looked at the anterior-most row of teeth on the dentary. To quantify tooth complexity, we used the method orientation patch count rotated, or OPCR for short. Allow me to break down what OPCR is and what it shows us. I'll start with the hypothetical, perfectly conical tooth viewed from a lateral perspective. In OPCR, teeth are analyzed from an occlusal or top-down perspective. In other words, imagine you're the prey item and you're looking down at the tip of this tooth. To assist with visualizing this, I now added a small black ring to the tip of the tooth. Using OPCR, the orientation of each pixel in a 3D scan is determined and then grouped as adjacent pixels that share a similar orientation in one of 8 patch colors. In our perfectly conical tooth example, viewed from the occlusal surface, this tooth would be composed of 8 patches, with each patch divided equally. Therefore, a patch can be defined as a group of 2 or more pixels with the same orientation. Let's look at another example, this time rodent molars analyzed in the original paper where this method debuted by Evans et al. In 2007. Again, OPCR needs to be teeth from an occlusal perspective, and so imagine you're looking at these molars as if they're prey item. As they are molars, their tooth surface is far more complex than the conical tooth, and so once OPCR is performed, we see that these molars have far more than 8 patches present. In fact, in their study, these molars were found to have 122 patches total. Given that molars are far more complex than cones, what this means is that a higher total patch count equals a higher tooth row complexity. So OPCR shows us the total number of patches in the tooth row, and that a higher patch count is equal to higher tooth row complexity. Applying OPCR to damselfish jaws, here we have a lower jaw of the damselfish viewed from the occlusal perspective. The OPCR output here shows us that the tooth row has 195 patches total. Patches are in part determined by resolution value, known as data rows per tooth, or RPT, in which a tooth is composed of a set number of data rows. In this example, the resolution is 40 RPT. For our study, we looked at each of our tooth rows at 4 separate resolutions: 15, 25, 40, and 50 RPT, with 15 being the lowest resolution, and therefore the output is more pixelated, and 50 showing the highest resolution, therefore the output is much less pixelated. We did this to understand how resolution influences complexity, as it could also affect the number of patches we're able to quantify. What we can see in this example is that patch count increases with complexity. In this specimen at 50 RPT, this denotes greater than 195 patches because the software used for the OPCR analysis crashed at this resolution, which is a fairly common occurrence at 40 to 50 RPT. Finally, we use 2 separate metrics to understand our OPCR outputs at each resolution level. The first is the OPCR total, which is the patch count generated for the entire tooth row, as what we see here displayed for each of our 4 different resolutions. The second metric we use follows a methodology in Maelstrom 2017, and it's called OPCR average. OPCR average is calculated by taking the total OPCR value and dividing it by the total number of teeth in the tooth row. So in this example here, we have 16 teeth in this row. We take 195 patches divided by 16, and that will give us the OPCR average value. And this value equally distributes the complexity along the jaw to each tooth in the jaw. Regarding analyses, for both our ecotype designations and the phylogeny used in our study, we use the most recent phylogeny by McCord et al. And followed ecotype designations provided in their study. Ecotypes are broad ecological functional categories composed of where an organism feeds and what their diet is. So for damselfishes, these ecotypes are benthic herbivory, which will be coded in green in our study, intermediate omnivory, which will be coded in orange, and pelagic planktivory, which will be coded in purple. To test our hypothesis that tooth surface complexity varies with diet in damselfishes, we examine group differences across ecotypes using Kruskal-Wallis tests, and for pairwise comparisons, we use Wilcox tests. Finally, to test for ecotype differences while taking phylogeny into account, we performed the phylogenetic ANOVA using the RRPP method by Adams and Calder. So for results, I'll start by showing you the relationship between the number of teeth and diet across damselfishes. On the x-axis, we have the number of specimens collected for each ecotype, and on the y-axis, we have the total number of teeth present on the jaw. In comparing all ecotypes together, we found the group differences in tooth counts across ecotypes is only marginally significant. When doing pairwise comparisons, tooth counts were only significant between herbivores and planktivores. So the big takeaway from these results is that we found there's only a marginal effect of ecotype on the number of teeth on the jaw. I'll now show you our OPCR average results across our 4 different resolution bins represented on the x-axis. That's 15, 25, 40, and 50 rows per tooth. On the y-axis, we have our OPCR average values, which you'll recall is the OPCR total divided by the total number of teeth on the jaw. First, starting in our lowest resolution bin, we see a similar pattern as before with tooth count, with herbivores showing the highest complexity and also showing a fairly wide range. Omnivores and planktivores show similar levels of per tooth complexity overall. Now looking at 25 RPT, notice we have lost 1 specimen from our benthic herbivory ecotype. It was 17, sampling is now 16, and this is due to the OPCR software crashing due to high tooth row complexity. Otherwise, you will see a very similar pattern at 25 as it did 15 RPT, with herbivores showing higher complexity than omnivores and planktivores overall. At 40 RPT, we now see that our sampling has dropped for all 3 ecotypes, but our patterns for each ecotype largely remain the same. And finally, at our highest resolution, we've lost over half of our total specimens due to the OPCR software crashing. However, still we see a similar pattern being maintained, with herbivores showing high complexity, while omnivores and planktivores show substantial overlap. Putting this all together, there are 2 big takeaways from these results. The first is that complexity increases with resolution, which isn't too surprising, and we can see this clearly in the general increase to each ecotype at different resolution levels that we use. The second and more encouraging result is that pattern of tooth complexity are consistent across resolution bins, with the median value of herbivore complexity consistently higher than either omnivory or planktivory. With this in mind, for the remainder of my talk, I will only show you results pertaining to our lowest resolution category, because it also has the highest sampling. However, keep in mind also that these patterns of complexity are consistent across resolution bins. So looking more closely at our OPCR average results at resolution 15, we again see the same general pattern of higher herbivore complexity and lower complexities in omnivores and planktivores. Applying a one-way ANOVA to our data, we find the average tooth complexities across groups do in fact differ significantly. Applying pairwise comparisons of ecotypes, we see that herbivores also show significant pairwise differences with omnivores and show very significant differences with planktivores. However, once we take into account phylogenetic relationships using the phyloANOVA, we find our significance drops substantially. In other words, differences in complexity due to ecotype are only marginally significant once phylogeny is taken into account. To understand why this might be, let's next take a look at our different specimens and their average OPCR complexity as they are distributed on the phylogeny. So here we again have our McCord phylogeny, and we pointed out the nodes to which matriculates the damselfish family originated. Our 50 specimen sampling represented here is about 12% of the entire Pomacentridae family. Plotting the tips of this tree are the average complexity per tooth for each species, color-coded by ecotypes. What we can see here is that herbivores overall have a fairly wide distribution on a tree with multiple origins, and they also have the highest concentration of complexity levels aggregated in a Microspathodontidae subfamily. Regarding omnivores, although sample size is smaller in those ecotypes, we can see that they are reasonably distributed on the phylogeny, and they have lower complexity overall. Planktivores, on the other hand, are the most common in our sampling with a wide distribution and also a wide range in complexities. Overall, it is clear that the aggregation of herbivores and the Microspathodontidae could be what accounts for the marginally significant results from our phyloANOVA. So, tying this all together, do our results support our original hypothesis that tooth surface complexity varies with diet and damselfishes? Our data suggests that yes, they do. Herbivorous fishes consistently showed the highest levels of tooth complexity, and they also demonstrated a surprisingly wide range in complexity. Regarding omnivores and planktivores, they show similar complexity, although planktivores do have a wider range. And finally, aggregation of high complexity herbivores in the Microspathodontidae could explain why herbivory loses its significance once phylogeny is taken into account. Regarding future work, how animals interacting with prey items may also affect patterns of complexity along the jaw. In mammals, higher complexity teeth are typically located posteriorly along the jaws. However, in damselfishes, we notice that higher complexity teeth seem to be located on the anterior portion of the jaws, differing from mammals. Running OPCR analyses on individual portions of the jaws will help to verify whether this pattern is quantitatively supported. And finally, reef fish are such an excellent exciting system to pose macroevolutionary questions and ideas too. So I look forward to further exploring patterns of dental complexity and its macroevolutionary consequences and other major reef fish lineages using OPCR and associated methods in the future. With that, I'd like to thank all members of my lab, both past and current, for listening to my ideas and hearing practice this talk. I'd especially like to thank Corey Evans and his lab at Rice University for access to training to that wonderful micro CT scanner. Also, special thanks to Keegan Melstrom for his generous guidance and advice on using OPCR analysis. I'd also like to thank all the awesome museum curators and collection managers for sharing their specimens with me for this study. And finally, I especially want to thank the Bell Museum of Natural History, the University of Minnesota, for the funding to pay for this research. With that, thank you again for coming to my talk. I look forward to any questions that you may have.